Hello everyone and welcome back to Genaura Academy, where biology is made simple and enjoyable. So today we are going to explore one of the most important ideas in biology, the central dogma of molecular biology. Central dogma of molecular biology. So if you have ever wondered how your body knows exactly what to do, like healing a wound, growing new cells, or digesting food, it all begins with the flow of information inside your cells. So let's understand this step by step. Before any protein is made, the cell must first copy its DNA, a process called as DNA replication. The copying of DNA by its own is known as DNA replication. This ensures that when a cell divides, both the cells receive the same genetic information. So this is the parental DNA and it is producing two new DNA molecules. It is producing two new DNA molecules and both the new cells are receiving the same genetic information they are receiving. This DNA replication, the nature of this DNA replication is semi-conservative in nature. So, semi-conservative in nature. So, as per Michelson and Stahl, he gave this nature of DNA replication semi-conservative in the year 1958. In the year 1958, Michelson and Stahl proved that the DNA replication is semi-conservative in nature. So what this semi-conservative in nature means? Which means that a new strand, each new DNA molecule contains one original strand and one newly synthesized strand. How? Say for an example, this is the DNA and when this is dividing, this is the parental DNA and it is the newly synthesized DNA. So it is containing one strand of the parental DNA and one of the newly synthesized strand. So semi-conservative nature means when a DNA is having one parental strand and one newly synthesized strand. Then it is known as the semi-conservative nature. Okay. How this replication is happening? So at first the enzyme helicase, the name of the enzyme is helicase. So what this helicase do? We all know that DNA is having two strands and the nature of this strands is anti-parallel in nature. Means if one strand is having its direction is 5 to 3 prime, so the another one needs to be 3 prime to 5 prime. And both these strands are joined together with a hydrogen bond. With hydrogen bond it is joined. These strands are containing nucleotides and these nucleotides are joined together with this hydrogen bond. So what this helicase do? Helicase enzyme breaks the hydrogen bond. It breaks the hydrogen bond. The role of this helicase enzyme is to break the hydrogen bond. So if this is a DNA, and it is having hydrogen bonds, hydrogen bonds. Once it will break this, then it will make a fork-like a structure called as replication fork. Means this part, the hydrogen bonds has been broken, but this is still having the hydrogen bonds. Helicase can't break the hydrogen bonds once at a time. So the process is slowly. Once it has been broken from this part, after that slowly it will break the whole. Up to here the helicase enzyme's function is clear. Now comes the DNA polymerase. So what DNA polymerase do? DNA polymerase has its two functions, its two conditions. Condition number 1 is it will work on single stranded DNA, and the second is it will function in the direction of 5 prime to 3 prime. So this is 5 prime, 3 prime, 3 prime, 5 prime. Okay, then add the complementary bases, and we know that A will pair with T and G will pair with C. So adenine and thymine is having double bond structures and in a double hydrogen bond manner, and guanine will add to the cytosine in triple hydrogen bonds. DNA polymerase couldn't able to understand that how it will start adding complementary bases. So here comes the RNA polymerase. The role of RNA polymerase is it will add primers. So here the formation is continuous. That is known as leading strand. But here what will happen? The fragment like structures forming. Say for an example, in this position 3 prime to 5 prime, 1 3 prime to 5 prime made. Now it has been broken from here. So it is blank now. Now it will again form a 3 prime to 5 prime. So these fragment like structures are known as Okazaki fragments. Okazaki fragments, and these strands are known as lagging strands. These strands are known as lagging strands. Then the RNAs will remove the RNA protein. RNAs will remove the protein on these Okazaki fragments. The primers which are being attached here, say for an example, the primers are helping them. So it will remove those proteins from the RNA proteins on the Okazaki fragments, and DNA polymerase will fill these gaps. These gaps has been filled by the DNA polymerase, and the DNA ligase will join the breaks and forms the continuous strands. So this was a brief introduction about how replication is. Now let's come to the main part, that is the central dogma. So central dogma is a concept introduced by Francis Crick. It has been introduced by Francis Crick in the year 1958, which describes the flow of the genetic information within a cell. Means from DNA to RNA to protein. From DNA to RNA to protein. DNA holds the instructions. RNA carries the message. And the proteins are the final product. They do all the real work inside the cell from structure to function. First step in this gene expression is called transcription, where a specific gene from the DNA is copied into the messenger RNA, mRNA. So this process, conversion of DNA to RNA, is known as transcription. It is known as transcription. So if you have already watched the previous video on the love story of the DNA and RNA, you might remember their relationship came with a cute condition. RNA must copy DNA's message but must use its own style. So instead of using thymine like DNA, RNA uses uracil. That's the special rule of their relationship. Means A complementary to U. This process happens inside the nucleus. This transcription happens inside the nucleus. An enzyme called RNA polymerase. So which enzyme is responsible for this transcription? RNA polymerase is responsible. Similarly, the way DNA polymerase was responsible for the process replication, like that for this transcription, RNA polymerase is responsible, which binds to the DNA and unwinds it slightly and builds an mRNA strand by adding complementary RNA nucleotides. A pairs with U and G pairs with C. Now comes translation, where the information in the mRNA is used to build a protein. The information built in RNA is used to build a protein, and this process is known as translation. This happens in the ribosome, which acts as a decoding machine. So the mRNA is read in sets of 3 bases called the codons, and each codon codes for a specific amino acid. For example, AUG. AUG is for methionine, which is considered as the start codon. So these amino acids are brought in by the tRNA. We all know that RNA, there are 3 types of RNA, that is rRNA, mRNA and tRNA. So these amino acids are brought in by the tRNA and linked together to form a polypeptide chain, which then folds in the functional protein. These proteins perform various functions, from making enzymes to supporting structures like skin, muscles, or sending signals like the hormones. This flow of information from DNA to RNA to protein is the fundamental or the foundation of the life. It explains how a single cell can grow into a human body, respond to the environment, and even repair itself. If any part of this process goes wrong, it can lead to diseases, genetic disorders, or malfunctioning of the proteins. So that's why understanding this central dogma is so important, not just in biology class but also in the medicine, biotechnology and genetics. But there is an exception. The exception is known as reverse transcription. Now, what is reverse transcription? In some viruses like HIV, or you can say retrovirus, the information flows backward, means from RNA to DNA, then RNA, then protein. So this process is known as reverse transcription, and for this process an enzyme is functioning. That enzyme is known as reverse transcriptase. Okay, so that's all about the central dogma. So far we have taken a bird's eye view of the entire central dogma, from DNA replication to transcription to translation. But don't worry, in the upcoming videos we will dive into each of these steps in detail. If you are excited, let me know in the comments which part you want next. And don't forget, the handwritten notes of the central dogma are already uploaded here in the channel. If this video helped you understand the overview, please like the video, share it with someone studying biology, and subscribe to the Genaura Academy for more content from class 9 to BSc level. I will see you soon in the next video, where we will break down the DNA replication in detail. Until then, keep learning and keep exploring life through biology. Thank you.